Hello, I'm a. Should I move this out of the corner? Oh, yeah. There we go. See if it I'm Michael. I'm a postdoc in Dylan's group. I did my PhD at the University of Wisconsin-Madison, doing atmospheric chemistry. I'm kind of continuing that. So my talk is about biosphere-atmosphere exchange of reactive carbon over a forest, a pine forest in Colorado, and these are measurements that I took this past summer as part of the flux exposure study. What?
Click on the screen once or twice.
Oh,
Now try.
okay. So what I'm going to be discussing is the surface atmosphere exchange of compounds attributed to atmospheric chemistry. So terrestrial ecosystems, forests can provide a source or sink of chemicals to the atmosphere through a process of emission or deposition, so flux. Knowing these flux terms and what contributes to them allows us to make predictions of atmospheric chemistry. Now I'm looking right at them. So just breaking down the different things that contribute to or global concentrations of species. So ecosystems can emit reactive compounds as a function of a bunch of different physical conditions of a forest, and I'm focusing on biogenic volatile organic compounds, these reactive carbon-containing compounds. So they can be emitted. These compounds can be transported to different places or react in the atmosphere via oxidation. So for example, isoprene and alpha-pinene are pretty common reactive BVOC, and they could react with these common oxidants, the nitrate radical, hydroxyl radical, and ozone. And they can make things like oxidized VOC, so just BVOC with oxygen. They could create aerosol or condense onto aerosol, and they could change the concentration and type of oxidants that are in the atmosphere. And then also soluble and reactive compounds that could deposit to the surface. So something like methanol or acetone or ozone, they could get solubilized by the surface or go into the stomata of leaves or other things. So thinking about all these things contribute to the global budget of an atmospheric species. So what we need are accurate and extensive field measurements of fluxes and concentrations of these reactive carbon compounds to predict atmospheric composition, air quality, and climate impacts. So just the top part thing was there. So what I do is, and our group measures these species using something called mass spectrometry, or specifically chemical ionization mass spectrometry, which detects species in the atmosphere based on their mass. So it relies on something called the reagent ion mixing with ambient air to make a charged product. So for example, we use something called PTRMS, or proton transfer reaction mass spectrometry, and we generate an H3O plus ion that mixes in the front of the instrument with our ambient air containing VOC, and then that'll make a charged detectable product, and it travels through the instrument and is guided to the detector that actually measures that mass or that product. And what a spectrum looks like is this. So if I were to sample, actually this is an actual spectrum of the forest. So what I get is all at the same time, pretty much instantaneously, all the masses of different chemical species that I'm looking at, and they make all their own peaks. And there's over like 1,000 peaks in the spectrum. So 1,000 different masses that are either compounds or parts of other compounds. And this instrument is, we like to use this because we could measure this wide mass range, as you can see. So all these different compounds have different stories. It's very sensitive. So what we're looking at are trace gases, so very low concentration gases. So we can detect them. And they're also selective in measuring the type of compounds that we want to measure, which is VOC. So I don't really have the time in 15 minutes to go through eddy covariance, but we use a method called eddy covariance to actually measure flux from a forest canopy, or the net flux. So we're taking advantage of the fact that vertical winds are transporting gases in these turbulent stochastic eddies. And what we can do is we could make a measurement of vertical wind and concentrations of gases at the same time and space in the same instant to gather flux of a species in or out of a forest. So what the actual equation is we're looking at just instantaneous deviations for mean or variance of 10 hertz measurements. So we measure over a 30-minute period, wind really fast, concentration really fast, and each time point we just take a deviation from the mean of each. But really all you need to know is that we can measure flux by these covariances and wind and concentration, and a positive flux is emission from your point, and then a negative flux is deposition or loss to the forest canopy. So we used flux to measure reactive carbon using two different mass specs. I'm just going to talk about the PCR mass because that's what our group used. We measured flux using two mass specs over forests in Colorado to measure the concentrations of biogenic VOCs and sources and sinks and such. So some of the questions are, what is the net flux of these key reactive species above a forest canopy? What does the vertical distribution and concentrations tell us about within canopy behavior? So if I were to measure at a bunch of different points within this forest, would I have different concentrations at different heights? And then how well does a chemical transport model actually reproduce the net reactive carbon budget? So we're making these measurements to be able to evaluate or train some model that can then, I guess, predict for a more global impact. So the location that we made this measurement was the Manitou Experimental Forest in Colorado, outside of Colorado Springs. It's a plantation that's mostly, actually it's all ponderosa pine trees with some shrubs, and it's a sparse forest, and its canopy height is about 15 meters. And what the experiment was that we made measurements along this tall tower, or this tower, not tall tower, this tower in the Manitou Forest, and we put inlets at different heights. So at each height here, there's an inlet sampling gas, and then at 3 of the heights at one time, we also had sonic anemometers to make that wind speed measurement. So that's what the little fork thing is. And the basic process was that we measured flux for 30 minutes at some point. And for the first and last week, we measured flux above the ground. And then for the rest of the study, we measured flux above the canopy because that's the bulk of what we're trying to get. And then for the other half hour, for every 5 minutes, we just cycled through each inlet that's below. So we're getting in canopy concentrations or gradients for 5 minutes each, and then we're measuring just the flux and concentrations above the canopy at 27.8 meters. So now I'll just go through some results because they're fun. So the largest reactive carbon source that we saw at the site was 232 MBO or methylbutanol. It looks like this. Here's some chemistry. It's commonly emitted from pine trees. It's very reactive against very common oxidant, the OH radical. And what's interesting to atmospheric chemists is also that it's, when you detect it, it's actually the same mass, it's the same charged product as isoprene, which is the world's largest source of non-methane hydrocarbons. So it's important to get both these numbers correct. Yeah. Also, it kind of smells like old celery. That's my smell. So this is so I'm going to show a few of these types of plots, and what it is on the top are the concentrations that I measured during the ground portion of the study. So when I had the 30 minutes at the ground, and then for the other portion of the study when I had the flux period at the top of the canopy, essentially. So that's concentrations on top, and then on the bottom are the actual fluxes. So concentrations, and then just either emission or deposition, but we're seeing that emission. So what we see for MBO that kind of sticks out to you is that we have this period of very high concentrations of MBO in the morning. And that's because in the morning, the surface is still pretty cool, so there's not a lot of mixing that's occurring. But we're actually getting sunlight and enough heat that allows our trees to actually emit MBO. So it's all just getting kind of trapped at the surface. It's happening. On the right, I'm also going to show deal profiles. So for the entire study, I just averaged each concentration over every hour. And then I also have that for each height. So this is just like the average picture of the entire study. And yeah, the top is concentration and the bottom is fluxes. And I have fluxes at 3 different heights because we were measuring concurrent fluxes. So we did have 30 minutes at the top, but we also had shorter periods below. So what kind of sticks out about this is that we have the strong gradient in the morning, MBO, which also drives a strong flux that we see. So we have a lot building up, but we also have a lot that's coming out of the canopy. And then we see later in the afternoon, we see emissions occur for MBO, and those are kind of correlating with surface temperature. So surface temperature allows for these plants to emit these compounds. And there's virtually no flux from the forest floor. So you can see it's kind of a huge step change. Like we see nothing, and then we see a lot of flux. So these are primarily the emitting from the pine trees. The next molecule that's the second largest reactive carbon source are monoterpenes. So there's a lot of different monoterpenes in this world, and they all share the same atomic mass. So we measure things by mass. They could range from highly reactive, moderate reactive, and pine trees also emit a lot of them, and they smell more like pine, less like celery. So this is what monoterpenes look like. So it's a bit different. So we see the peaks in monoterpenes is in the evening time when we're actually we're just building a lot of monoterpene in that low mixed space. That's pretty cool. And then on the bottom, we see emissions of monoterpenes that are not as exaggerated. And in the deal profiles, it's just reflecting what we're seeing on the left. So we have this evening, early morning buildup of monoterpenes. And then we also see emissions of monoterpenes peaking with surface temperature. And then we still see emissions of monoterpenes near the forest floor, but it decreases. So there's a larger source from what you're measuring above the canopy than if you're measuring closer to the ground. And then this is a lot of stuff, but this is going to be up for maybe a few minutes. So I just showed the two largest sources, but we actually have like 500 noted reactive carbon compounds. And I'm just listing a bunch here because these might be some that you're familiar with and are very ubiquitous compounds. So methanol, acetone, ethanol, and acetaldehyde. You can see that they all are net emitting over the canopy. So they are soluble and potentially might deposit, but we actually have a large enough source from the plants that they would be emitting these small OVOC. And then you could also see near the ground that we had higher emissions of these OVOC near the ground at the beginning of the study when we had a lot of nice greenery on the ground. And then when we got to the drier portion at the end of the study, when we went back, we didn't have that. And we saw this decrease in emissions occur. So greenery on the ground actually contributes this net emission of OVOC that we're seeing. And then looking at these concentration deal profiles, you can see that they all kind of have these unique profiles that will tell you something about the compound that we're going to delve into more, not in this talk, but just are interesting. So for methanol and acetone, we see this morning peak occur that was similar to the MDO that we saw. For ethanol and acetaldehyde, boxing things, we can see that it potentially has more of a ground source based on, it's hard to see because it's kind of colored, the color scale is off, but we have more of a ground, higher, close to the ground concentration of ethanol and acetaldehyde than later. And then methanol and acetone show this buildup throughout the canopy, so it could be more of a tree source. And then finally, part of this project is to take these values that I measured and kind of put it in the context of a global model and see if the global model that our group uses is getting this area right. So what I'm using is Geoschem. It's a 3D global chemical transport model that handles transport of gases, emission deposition, reactions. And what I have on the right here is just an example model input of MBO emissions and showing where our grid cell is. So the results that I'll show are just for the one grid cell that contains my site, pretty much. And what I did here is just I added up all of the reactive carbon flux for the whole study and made a deal profile and compared it to what just the base case of our model looks like. And we can see that we're under predicting the total reactive carbon emitted over that study period. But a large portion of this is because of the land cover that was used in this model, because we were, our footprint is actually, our footprint is actually a very small part of whatever the grid is that we're putting our land cover into. So we have to get a little more specific. So on the left is what the model thinks that our footprint should be containing, because it's really just a larger grid cell. But what we really see here are a bunch of trees and grass. So I took the image and just counted how many trees there were and took a land fraction based on that and just said it was either ponderosa pine or grass. Then when you do that, and then you reassess, redo the entire model, you can see that you actually get closer to the values that we're measuring, and it kind of does better overall throughout the whole study, get closer to the reactive carbon that we're seeing. And I think that's it, yeah. And then for acknowledgements, these are all the groups that were involved in the study. So our group at Twin Cities and Colorado State and University of Indiana, and this is NSF study. Yeah.
I have a question about spatial variability. I'm on the soil side of things, so I don't see much of atmospheric carbon emissions. But I'm wondering, when you're talking about that grid cell size, to what degree, like, what is your cell size that you look at when you take a measurement? Are you looking at like
It's much smaller. Yeah. So.
Or does it have to be more like species or land cover?
So right. So the grid cell that I'm comparing is probably about 100 times larger than my footprint, like the actual measurement footprint that I'm seeing. So I guess that's what was your question, I guess?
So I guess I'm wondering, like, what, when you take a measurement, how much land area does that represent?
Oh yeah, it's like 0.01 square kilometers, I guess. It's like 100 by 100 meters, pretty much for the bulk of the flux. And then we have a 0.25 or 0.025. What's the numbers? 0.25 by 0.3125 degree grid cell. So it's much larger.
Michael, can you hear me?
Yeah.
Yes, this is Paulo Pagliari. I'm a professor in the department, and I do nutrient management. I have a study where I have been collecting rainfall and snowfall from every precipitation throughout the state. And I found quite a bit of organic nitrogen that comes down with the rainfall and snow. Do you know what organic compounds could contain nitrogen that are suspended in the atmosphere?
What organic compounds that can contain nitrogen?
Yes.
Organic, like in that they're carbon containing as well?
Yes.
So there are things like, I don't know if they're contained in snow, but like isocyanic acid, HNCO, just common biomass burning. And then there's a lot of organic nitrates that can be produced. But I don't really think of that in terms of things that are scavenged by snow. I haven't really thought about things like that. But how do you measure the organics, I guess?
So what we do is we measure the inorganic ammonium nitrate, and then we do a autoclave digestion with persulfate digestion, and then we run the nitrogen again. We run the ammonium again on the sample, and then by difference we assume that is organic. And it's quite a bit. We are seeing like between 15 to 20 pounds per acre per year being deposited in that particular form. It's quite a bit.
You can have aerosols. I guess these mixed phase aerosols that then surface reactions can occur on them, and then you change this. Then you could make more organonitrates that way. But that's
Okay.
Then,
is there any method that you can suggest that we could try to investigate what's in, what forms they are?
Of speciating?
Yes.
I guess GC is the only thing I can think of. But I guess you'd have to liquid phase extract them or LC. Some type of chromatography.
Okay, great. Thanks, Michael.